Hi, in this video I'm going to introduce you to the Dialogue.bio agent's dialogue system, and we're going to try to solve an example problem together. So first let me show you the interface. On the right hand side you see a chat box and you see Bob is connected and said tell me what you want to do now, so we can start talking with Bob by entering text into this box and either hitting enter or pressing the send button like this, and you see Bob answered. The little checkbox here means that Bob got this message. In one condition, you will be talking with Bob such that a wizard is also connected to the room, another human. And if you ever want to say something that you don't want Bob to try to interpret, you can click the wizard mode and say something that will not be picked up by Bob, and you see that the little tick mark is missing, meaning that Bob didn't see this message. And when I uncheck this, Bob will start answering again. A few of the other things you can see on the interface here is a reset button. If you ever need to reset the conversation or just start a new model, you can press this button to do so. You also see a number of example sentences listed here. You can use this in addition to your cheat sheet as a help in finding relevant things to ask to solve your problem. And when you click any of these, that text gets copied into the chat box, so you can just hit enter to send it. On the left-hand side, you see a currently blank canvas that will visualize the model that we built together during the dialogue, and the model will represent our hypothesis about a molecular mechanism that we are discussing. So now let's look at an example problem that we will try to solve through dialogue with Bob. So here, assume that this is an experimental observation that SB525334 decreases the amount of SMRF2. And so we are going to try to talk with Bob to find relevant information, formulate a hypothesis as to why this might happen, and then describe a model of the relevant molecular mechanism, and finally check whether that model can reproduce this observation. All right, so let's start by asking what this SB525334 might be. Okay, so it is a drug, so we might ask about its targets. All right, so it looks like the nominal target of SB525334 is TGF-BR1. So we can, in fact, start building a model of this process by saying, let's build a model. And now I can describe the mechanism that I want to model. In this case, I want to say that SB525334 inhibits TGF-BR1. Okay, so Bob interprets this mechanism and creates a mathematical model whose visualization we can see on the left-hand side here. Sometimes it can take a little bit of time to generate the layout of the model, and you can press this button called Perform Layout to speed that up. And you can actually zoom in and out on this interactive canvas. You can drag the canvas to pan, and you can drag each of the individual nodes on this canvas. In addition to this view, where you see reactions displayed as, for instance, the drug SB525334 catalyzing a reaction in which the active form of TGF-BR1 transitions into an inactive form. In addition to this view, you have other views, such as the contact map view that would show any binding between proteins of interest. Here we don't have any. The influence map, where you see processes and how they relate to each other. We will come back to this later when we have a more interesting view here. And the reaction network, which is similar to the basic network view, but it's a static image, and it also displays a little bit more detail in terms of the states of different proteins and the reactions between those states. So let's switch back to the network view here. Okay, so now we need to find out how this effect on TGF-BR1 ultimately leads to a decrease in the amount of SMRF2. So let's see if we can find something that regulates the amount of SMRF2. Okay, so it looks like there are several transcription factors that regulate the amount of SMRF2. So see if TGFBR1 activates any of them. For instance, SMAD2, which I happen to be familiar with, so I prefer to ask about that one. All right, so it looks like there are several different types of relations that connect TGFBR1 to SMAD2. And you see that in this case, Bob gives us an answer, but it also tells us to look at the provenance tab to see additional information. So I can click on this provenance tab. And what I see here is that there are 61 pieces of evidence for the fact that TGFBR1 activates SMAD2. And in fact, I can click this link to get to a page where I can browse the literature evidence for this relationship and decide for myself whether I think that this is a plausible mechanism. So I do believe that this is probably the right explanation here, so I will keep building my model and say TGFBR1 activates SMAD2. Okay, so what happened here was that the model was extended according to what I said, which is that TGFBR1 activates SMAD2. However, the computer alerted me that required activity is possibly missing. Namely, I should have specified that TGFBR1 has to be in an active state to activate SMAD2, because currently, whether it's inactive or active, it will activate SMAD2 in both of those cases. You can also diagnose this issue in the influence map, where you see that there's no relationship between the process of the drug deactivating TGFBR and TGFBR activating SMAD2. So what I can do here is that I can take this suggestion and just add it to the model. And what happens here is now we have specified the activation of SMAD2 by TGFBR1 to only apply to the active form of TGFBR1. We can go back to the influence map, and we now see that the drug deactivating TGFBR1 has a negative effect, a red negative arrow, to the ability of TGFBR1 to activate SMAD2. All right, so this seems good, and then now we can bring in the final piece, which is the amount of SMRF2. So active SMAD2 increases the amount of SMRF2. All right, so now this has been added to the model. So we see our path from the drug all the way to the production of SMRF2. And so now we can ask questions about this model to see whether it satisfies the properties that we are looking for. So does SB 525334 decrease SMRF2 in the model? Yes, it is decreased. And in fact, whenever we ask about model properties that are analyzed through simulation, we get a tab here called Sim that we can click on to look at the simulation results. And we see the simulation results for the amount of SMRF2. So without condition means without the drug, it keeps increasing. And with condition, that is with a high amount of drug, it stays effectively constant. So that means that the drug negatively regulates the amount of SMRF2, which is exactly what we wanted to explain. So a few other interesting pieces to the interface here. First of all, you can resize the dialog box and also the canvas, so you can fit it to your screen. Make sure that you are looking at the screen in a way that you see all these tabs, so you can switch between them and look at whatever you find relevant. And there's also a note tab here. So if you ever want to keep track of some information, for instance, from the provenance tab, like let's say I want to make sure I don't forget that TGFBR1 activates SMAD2, I can copy paste some information here. I can also take some other notes. So you can keep track of information here as you are solving the problem. All right, so that's about it. Good luck and have fun trying to solve the tasks.